platform to support understanding of pesticide use and its impact on human health and the environment. And Jakub will guide us through what the SPRINT Toolbox is, why it was developed, and how you can use it. So very interesting stuff today, I would say. So again, welcome everybody. So note that this is a webinar, so you won't be able to switch on your microphone or open your camera. But we do encourage you to make use of the chat, where you can post questions in the Q&A session, but also there Charlotte will post relevant information about the SPRINT project. And we also love to have some interactions there to see, well, who is listening, why you're listening. So do make use of the chat. Just for your information, this webinar will also be recorded and will be placed on our YouTube channels. But yeah, because no one is visible besides the speakers, we thought this would be okay. So without further ado, we are going to our first speaker, and I want to give the digital floor to Violette Geissen of Wageningen University. Violette, take it away.
Yeah, Paula, thanks for the presentation and the introduction. I'm really happy to present now the final outcomes of the SPRINT project. We started the SPRINT project nearly 5.5 years ago, and now it's really up to its end, and we had very, very interesting results. The topic today is pesticide residue mixtures detected in ecosystem and human matrices across Europe. I think we presented it already sometimes, it's as well published. But today we will focus on what is the actual risk for ecosystem and potential risk on human health, and what do we recommend as new tests and as well for EFSA or policymakers. But let's go back to our field campaign conducted in 2021 in 11 case study sites, 10 in Europe, 1 in Argentina. And we sampled here all ecosystem matrices, environments from the terrestrial and aquatic ecosystems, and as well more than about 670 participants, farmers, organic farmers, neighbors, and consumers were participating, and we tested more than 200 different pesticides. And some were used as biocides and a synergist. As you can see here in details, which samples were collected. So from the soil and water bodies, plant samples, outdoor dust, air, and so on. But as well, we collected indoor dust, and from humans, farmers, neighbors, consumers, many nasal swab, urine samples, blood samples, stool samples, wristbands were sampled, and as well, we conducted a DPA analysis to see what is the dietary intake.
Violette,
so in total, yes?
Just a small thing, your slides are not changing.
Oh, that's strange. Does it change now? That's really strange.
I don't see it moving, but maybe others.
I think
no, the chat is
maybe
also
I stop
saying...
sharing and share again. Maybe then it's functioning.
Yes, let's try.
It's working now?
Now we can just see your normal screen, so it's not in presenter mode.
Now?
Yes.
You see now the second slide with environmental samples and human?
Yes.
Great. Okay. So the question is now, we asked how many pesticides are applied in each case study sites. So we saw that 7 to 37 different compounds were applied in each case study site. These case study sites were covering the 10 main cropping systems in Europe. And following the farmers' information, 23% of the applications exceeded the recommended application rates. What did we find there? I think that is already known by most of you. We found in all components Here presented soil, crop, air, indoor dust, and water and sediment. For organic farms and conventional farms, we found overall mixtures with a high number of pesticides in the middle of the growing season. Organic farms related ecosystems had lower concentrations and as well lower numbers than the conventional farms, but this was not meant for air. And what the most interesting thing for us was that we had a strong accumulation in indoor dust, as well for organic farmers as for conventional farmers. And here's a top row with 123 different pesticides. What did we find in air, in urine and feces? Here as well, and the Netherlands showed the most numbers, the highest number of pesticides in urine, until 16 different pesticides for conventional farmers. For feces in Argentina, we could see them, and in Italy, we could see the highest concentrations and numbers as well of pesticides. So what does it mean? Does anybody know what does it mean for human health? So that was the main question we had here. And we here in the SPRINT project, we were working with the pre-market assessment and the post-market surveillance, which is now strongly reduced following the last changes in the legislation. But we will show you here what we found and what we can, how can we convert the outcomes we had from the case study sites to risk. What does it mean for ecosystem and human health? First of all, I will show you how we did with ecotox tests we were conducting. Based on the findings in the case study sites, we prioritized mixtures for ecotox tests. And what we did here, we took all the lists of pesticides detected in the aquatic or terrestrial system. We classified them by risk coefficient and frequency of occurrence, and therefore we ranked then the 5 most important pesticide residues per case study site and came up with 11 different mixtures we tested. For the terrestrial ecosystem, we added as well here persistence. So it looked like this. For the terrestrial ecosystem, we were analyzing, taking a lot of indicators into consideration, as well for the aquatic ecosystem. So we were here working with EFSA indicators, but as well with new indicators like wild bees, like behavioral changes, and so on. I can show you here an example for the SPRINT aquatic ecotox tests. Here were ecotoxicological parameters taken beyond core endpoints, suplatal and functional endpoints, biomarkers, cellular and tissue level effects, trait-based and life history metrics, population level parameters, and community and ecosystem endpoints. So we extended a lot the standard EFSA parameters which are used. What came out? Here looking at the aquatic and terrestrial ecosystem, and here we show the number of pesticide mixtures from the 11 mixtures which we have taken, for which there was shown an effect of more than 50% at predicted environmental concentrations, or for the terrestrial ecosystem, predicted dietary exposure quantity and application rates. And these are the values which by EFSA are said to be safe. So what we can see here is when we take different numbers of organisms taken into consideration for this risk assessment, we see in this, you can see here in the last column, the more organisms we take into consideration, that means that we test in mesocosms, the more of the mixtures show effects above 50%. So if we only look at algae, for example, none of the 11 mixtures were showing any negative effect. But if we take here different organism groups together, then 8 for the terrestrial system, 9 of the different pesticides mixtures tested showed negative effect on the ecosystem. What does it mean? We highly recommended shift to mixture-based assessments, set mixture thresholds, so it is very important that regulatory authorities should establish safe environmental concentration standards for pesticide mixtures that are likely to co-occur and addressing the combined effects, and these as well for the terrestrial ecosystem. It would be important that we focus on a set of native species because native species reflect more realistic ecological impacts and address bioaccumulation risks and enhance field monitoring and conduct retrospective environmental risk assessments in critical agricultural areas. So these are our main findings related to the ecotoxicology. What about human exposure? We have here to distinguish between dietary and non-dietary exposure. The non-dietary exposure can be done by dust ingestion, inhalation, dermal contact or dermal contact with air. And the question is, how high are humans exposed by dietary and by non-dietary exposure? And here it should be said that the EFSA criteria in this moment and the EFSA tests are only considering dietary exposure. And the question is for us, or was in SPRINT now, how high is the non-dietary exposure? But first of all, let's look how high of what amount of numbers we are taking up per day by our diet. So therefore, we made a double proportion analysis. Participants from these different countries, you can see here, from farmers, neighbors, and consumers. And then we analyzed the consumed food. The half was eaten, the other half was taken to the laboratory to analyze pesticides. And then we found the following results. The consumers, here shown with the points, the dots, it's what was taken in by diet. And with the triangles, you should see what is resulting concentrations in urine. We showed it here for consumers, farmers, and neighbors, conventional farmers, and organic farmers here taken into consideration as well by consumers. What we saw that we have an uptake of 15 to 39 pesticides per day, and that people eating organic food for the consumers have a lower intake than others. And then in urine, we saw numbers until the highest from as well the conventional farmers until 14 different pesticides in urine. What has to be taken into consideration that until now the maximum residue levels for pesticides are only defined for a single pesticide. So if we take up by dietary uptake per day up to 39 different pesticides, the risk for the consumer's health is not defined. And we don't know how high it is. We do not know whether the sum of all of them is still safe or not. Now let's look on the non-dietary uptake. And here we saw that the indoor dust as a source of non-dietary uptake is the main source of non-dietary uptake. In the indoor dust, as I showed as well in the beginning slide, in organic households, the main number is 51 to 75 different pesticides. And in conventional ones, it is the same between 51 and 75 and 76 to 100 different pesticides. The question for us was, are these pesticides which are detected in indoor dust, are they safe? Are they of hazardous concern? And therefore we took here, compiled the EFSA conclusions, seeing that a lot of the pesticides we detected in indoor dust are sure or possible cancerogene, endocrine disruptors, or causing reproduction development effects. It was more stronger for the non-approved pesticides than for the approved ones. But still, for the approved ones, we can see here, for example, for reproduction development effects, it's more than 60%, as well as in organic farming as in conventional, are meant to have negative, yes, or possible negative effects. It has to be said that it is not risk, this is hazard. That means this is the characteristics of the pesticides, and to define the risk, we need concentrations. Our colleague from Utrecht, they analyzed the number of pesticides detected in wristbands after 1 week of exposure. Wristbands are silicone bands which you wear 1 week in your wrist, and then pesticides are detected after 1 week. And here we saw that conventional farmers had 9 to 74, organics 8 to 66, neighbors more or less the same, and consumers 2 to 54 different pesticides in the wristbands. That means these were the pesticides they were exposed to via the air, via indoor dust, via non-dietary exposures. Then our colleague was calculating what is higher, the dietary exposure or the non-dietary exposure. For most of the pesticides, which were in low concentrations in indoor dust, they've seen that the dietary exposure is much higher and contributes nearly 99% to the total intake. But for glyphosate, it was different. Glyphosate was detected in high concentrations until 5 milligram per kilogram in indoor dust samples. And here, Daniel, he calculated the ingestion by dust and dermal contact based on concentration from farmers' houses and compared them to calculated dietary intake of glyphosate. And the dust ingestion here is shown in red and the dermal contact in blue. And the box plots, they show the dietary intake calculated. So what is clearly shown here for the different fruits and vegetables, that the non-dietary intake can be much higher than the dietary intake. And this is a very important outcome of the SPRINT project, as a non-dietary intake is actually not taken into consideration in the EFSA risk assessment and should urgently be added. So the key messages here is cumulative risk assessment performed for pesticides in food seems to be translatable to aggregated exposure. However, there is a big gap on available data from tox studies to derive noise for different combination of pathways and endpoints. For realistic worst-case scenarios, our model calculation predict that mixture exposure may surpass the acceptable daily intake. But we do not know in this moment how often this may occur. The duplicate portion analysis accounts for food processing and portioning, hence providing a more realistic data to calculate uptake of pesticides. Non-dietary uptake can be higher than dietary uptake and should be included into risk assessment and be considered related to maximum residue levels. It would be important to implement an EU-wide human monitoring program to identify really integrated exposure from all sources of pesticides. Expand environmental monitoring programs for air and the domestic environment of farmers' families, especially for indoor dust. Harmonized national registers of pesticide uses are required. Jakub will tell more about this. And targeted reduction in highest health risk pesticides and cropping combinations should be taken into consideration. Now let's move to human toxicity. Here a tiered approach was used. So tier 1, we assessed all the available data and in silico modeling. Then from this, we consider the 12 the 20 components of concern based on occurrence and hazard coefficients. In tier 2, we did in vitro testing, top 10 components of concern, and the in vivo testing were then done for the top 3 components of concern, and we did an additional test with the 13 most occurring pesticides. What are the key messages? And what did we do? Standard and new in vitro models were used to test the top 10 pesticides and mixtures of concern, showing cytotoxic and genotoxic effects in a linear and additive dose response, along with alterations and synergetic effects of kidney cells and organoids, viability and transcriptome. These were the new SPRINT indicators. Standard and in vivo models were used to test the top 3 pesticides and their mixtures, showing sex-related effects on endocrine disruption, liver toxicity, microbiome, and behavioral alterations and kidney toxicity. And important is to mention here that these effects were observed at doses that are currently considered safe. I will just give you one example here from our innovative indicators for behavior and gut microbiome. That was conducted at the University of Cork. So what we can see here, mice were exposed 15 weeks to glyphosate in a different dose, control acceptable daily intake, 10 times acceptable intake and 100 times acceptable daily intake. What was very interesting to see is that glyphosate-driven effects on anxiety-like behavior is already seen at the ADA level. As well, they could see glyphosate-driven effects on cognition and general sociability below the Noel effects. These are normally concentrations which are considered actually to be safe. Because we used new indicators and looked at the behavioral changes, we could find significant differences here in the mice experiment. Furthermore, it was clear, and this was confirmed in all the other tests we conducted as well with rats, that microbiome structural shifts are very clearly caused by glyphosate exposure. And what our colleagues as well did, they did a fecal microbiome transfer to healthy mice, and they as well led to a transfer alteration in social behavior in healthy individuals. That means the conclusion is that we can link microbiome community changes and social behavior. We additionally did, because we find so high concentrations, so high numbers of concentrations, we did an additional project experiment with our colleagues in Antwerpen. And they tested, after 15 weeks, that's 15 weeks long as well, the 13 highest at-risk pesticide mixtures below the acceptable daily intake. So that means that was so no effect would be seen. But what they found is that they as well saw behavioral changes in female rats. They saw in the male rats decreased flexibility of the blood vessels. The microbiome as well confirmed changes, and kidney functions still changed, it still functions, but we found significant structural changes. These very sensitive indicators show that even if we are exposed to, or the rats are exposed to concentrations above the acceptable daily intake, they may cause small changes in the body, and we do not know which health effects this will cause on a longer time. So the key messages for human toxicology: the new models used, both in vitro and vivo, provided to be sensitive and relevant for different ecological, toxicological endpoints of interest. The in vivo information provided by the new SPRINT indicators on behavioral effects is mature to be used for risk assessment and pre-market evaluation. The in vivo mechanistic information provided by the new SPRINT indicators on microbiome needs further validation to relate specific health risks to microbiome alteration. The in vitro mechanistic information provided by the new SPRINT indicators could inform hazard assessment on different endpoints, in vitro kinase and organoids viability and transcriptome, but further validation is required. I will skip this and then come to a very important issue as well, which I did not mention until now. Our colleagues from DTU in Denmark, they calculated damage costs per year related to ecosystem quality and human health. They used here the UZ-OX model for. And as you can see, if we look here only at the dot, the blue dot, that external costs are summing up until 8 billion euro, for example, here in Italy, and so as well in France. So these external total damage costs should be taken into consideration if we try to come up with economically viable agriculture, because the farmers, we want food which is cheap, but the external costs are paid by the total society and not only by the consumers. These are hidden indirect costs. We have to pay for cleaning of drinking water, human health costs, etc. So the take-home message is pesticide residues mixtures are omnipresent in ecosystem and humans, highly accumulated in indoor dust. Most of the residues are hazardous for ecosystem and humans, and the real risk is not considered in this moment and not known, especially of the mixtures. We need pesticide regulation that assess the real risk, including sensitive indicators. We need a transition to a more sustainable and 2030 ready agriculture systems based on agroecological concepts. And all stakeholders need to be on board to achieve a transition towards reduced pesticide use. Thank you for your attention, and please feel free to put your questions into the chat.
So thank you, Violette, for your presentation. There was already one question posed in the chat, and that's from Stefan Robinson. He asked, would bioassay be a realistic approach to assess impact by pesticide mixtures and avoid long tests with various animals?
The thing is that if you only use, for example, in vitro tests, you can only assess the hazard and not the real risk. So if you want to really know what is going on in an organisms, in a whole organism, then I think it is difficult to not use in vivo experiments.
Okay, yeah, thanks. I don't see any other questions. Yeah, Stefan says thank you. Charlotte, you have your hand up? No? If not, then I think we should continue. Well, thanks again.
Paula wanted to add something which I forgot to say. I would like to thank all my colleagues, work package leaders and other colleagues who contributed to this presentation. I didn't put them all to not take more time, but thanks for all contributing with the slides.
Okay, there's still one question. Has EFSA reacted on your outcomes?
We had a meeting with EFSA 1 year ago and we discussed our outcomes, and they are really interested in our outcomes, and hopefully they will take some of our recommendations into their advanced risk assessment.
Okay, and then one final question is, recently There was a retraction of the Williams et al. Paper. What do you think the impact might be about the agrochemicals?
I cannot answer this question now. I have to go deeper into these facts. I didn't follow this process in detail.
Okay. Well, thanks a lot. Then we go to the second presentation, which I will give myself. So I will share my screen, and please, if someone can say if they see my screen, that will be super nice.
Yes, we can see it.
Okay, thank you so much. So this presentation, is from Virginia, Aparicio. However, due to a language barrier, we decided together that I would give the presentation, but all credits are for her. Everything is for her. I'm just the translator, so to say. So I will start. So this presentation will show some of the Argentina SPRINT results of the fieldwork and some findings and perspectives. So, she also would like to emphasize that it's of course not only her work, but that the whole team was supporting her, and that she's very grateful to have this team to do this important work. Yes, so Argentina contributed to the SPRINT project by offering a unique agricultural context with large-scale farming systems that are important worldwide. Using the SPRINT protocols in this setting helped to test the approach in real high-intensity production conditions. At the same time, study both rural and urban population within the same system gave the project a broader and more complete perspective. So in Argentina, more than half of the farmland is planted with genetically modified crops. And most of these crops are designed to tolerate herbicides, mainly glyphosate. Only wheat is being the exception, the main exception. So Argentina's agricultural production relies heavily on pesticides, and glyphosate is the most used pesticides, and it makes up about 55% of all herbicide used in the country. Glyphosate is used across large areas of farmland nationwide, meaning it's present throughout much of Argentina's production system. The fieldwork that Argentina executed that followed the same biomonitoring protocol as the European SPRINT Consortium. So it uses the standard protocol that was published in +1 by Vera Silva to measure pesticide residues in a combined way in people, in their surroundings, and in the related production systems. And well, the key point here is that they use the same methods, and that makes that Argentina can be directly comparable with the other data from the SPRINT EU countries. So one of the main findings from the Argentina study was that people and the environment are exposed to a mixture of pesticides and not just a single substances. So they found several pesticides at the same times, and well, it's often called cocktails. And this is not unique to Argentina. Actually, in a whole SPRINT, it was clear that it confirms that the real life exposure is complex and it involves many substances at once. So the real exposure scenarios are inherently multi-phased and complex. So another important finding was that we could map how pesticides spread in the environment. We found found pesticide residues indoors, especially in household dust, even in homes that were not next to treated fields. So they travel through air or to dust. And this shows that these substances can move through the environment. It also highlights why SPRINT approach looks beyond food and water and considers the full living environment when assessing exposure. So then next thing is that they analyze the indoor dust in rural homes and they find pesticide residues inside the houses. So this is important because it shows that pesticide contamination is not limited to farms or fields, but also reach our own homes. These findings point to long-term, everyday exposure, especially for rural families, and highlight the need for integrated approaches like SPRINT. So when they looked at indoor dust in non-rural homes, they find a similar number of pesticide residues. This means that the level of contamination was not simply linked to how close the homes were to agricultural fields. This challenged the idea that exposure drops just by moving further away from farming areas and shows that why SPRINT looks at many different exposure pathways, so not only the direct contact with pesticides. And to better understand personal exposure, in Argentina, they also used the silicone wristbands. So they were worn by volunteers for 1 week. And these wristbands act as a passive sampler and capture what people are exposed to in everyday life. They analyzed almost 200 pesticides and detected 80 different substances, showing that people are exposed to complex mixtures. Although glyphosate was not included in the measurement, the main results clearly support the main goals of the SPRINT project. So this figure shows how variable and complex the real-life exposure is, even over a short monitoring period. It helps to illustrate that exposure is not constant and involves many substances at the same time. So what is the scientific legacy of Argentina participation in SPRINT? Well, first, it created a unique set of data. So for the first time, Argentina had a solid baseline generated using international standards and comparable at the global level. And second, it helped build a technical capacity by training people in modern integrated monitoring methods. And third, it provided a strong scientific foundation. This work does not express opinions, it produces reliable evidence to support informed discussion about future production systems. Argentina data sets strengthened also the SPRINT Toolbox by testing it under real large-scale conditions, and this makes to help the toolbox more robust and also globally relevant. So Jakub will tell more about the toolbox in a few minutes. So to conclude, projects like SPRINT show that the food production and health protection are not opposing goals. The key is using science to connect the two. Argentina's contribution helps strengthen that connection by providing real-world evidence. These findings are not the final answer, but a strong starting point. The invitation is to see the evidence not as a problem, but as a guide, supporting a constructive dialogue, better policy, and a fair science-based transition to a more sustainable way of using agrochemicals. So this is the last slide of Virginia, where she has her acknowledgement, and she really wants to thank all the people that helped her, especially the volunteer participants in Argentina. They were involved in the case studies. They were open to transfer a lot of material. She wants to thank the SPRINT Consortium and, of course, her team for helping her. If you have specific questions for Virginia, please contact us via sprint@word.nl, and we will make sure that you can keep in touch with Virginia. So this was her presentation. Thank you for your attention. I see a question. Can we find both presentations on the SPRINT website? Yes, everything will be published in a YouTube video where you also will find the presentations in it. So I will stop sharing. Okay. And we're back. So I have to switch roles now. So next up is Jakub to tell us something about the toolbox of SPRINT. So if you would like to share your presentation, then you can take it away.
Yes, good afternoon, everyone. I would like to use this opportunity to present to you the SPRINT Toolbox, which we really will help to shift the policies and strategies towards the sustainable use of plant protection products. It's the effort that has been done by many experts within the SPRINT project. So what is the toolbox? So the toolbox is actually not just one tool, but it's a collection of different tools, and the aim of the toolbox is to integrate many things together, actually. So both already available data, existing data that are existing in different countries, like pesticide registries, pesticide statistics about pesticide use, but also the results that were measured in different SPRINT activities, and combine that also with the literature data, with models and estimates. And what kind of data we were interested in? So we were interested in all the data related to pesticide risks, pesticide exposures, pesticide hazards. So that means that we focused on pesticide exposure predictions, pesticide exposure modeling, and collecting and visualizing the effects of the pesticides and pesticides mixtures, both on ecosystem endpoints and human health related toxicity endpoints. We believe that all that results in kind of integrated assessment where we placed all these outputs in one place, and we also showed the way how to use such combinations of data to model and map the exposures and risks of pesticides and pesticide mixtures. And it was also done at different levels, so from local field level to national level to pan-European level. We really believe that after this mixing and after this integration of all these data, we are providing a set of tools that can be really helpful to several stakeholder types, policymakers, decision makers, but also a wide spectrum of scientists and educated public. And especially the target group of policymakers and decision makers can take this information to create the informed decisions and to prepare the pesticide reduction strategies. So technically what it is, the toolbox is actually a website where all the tools that we created are shown. And this website, you can see already the address where the toolbox will be available in a long term. And now it's password protected because it's still under final tuning. And in February, yeah, I can say sharply, on 15th February, there will be the Toolbox launch webinar where the Toolbox will be open freely for the wide public. And we are doing this last tuning also based on the feedback that we are collecting from different types of stakeholders who were testing the Toolbox, and they provided very good feedback, and we are now incorporating all these ideas in the final version of the Toolbox. So I prepared, due to limited time, just several examples what people may found in the Toolbox. So the first example is that, as Violette mentioned today, within the SPRINT there was very large sampling campaign in 2021, and all the results that were processed also into the scientific papers, but they were processed in a user-friendly way to be displayed in the Toolbox. So I can switch now to the toolbox itself. And there is a part which is focusing on these case study results presentation, where you can choose between the matrices. So you can have a, let's say, matrix dependent view on the results, showing what were the results of pesticide concentrations and numbers of detected substances in different countries, comparing the different types of farming systems, showing the same results as were published also in our scientific publications, but I would say much more easy to understand way. And it's interactive, so you can also click on the country you are interested in, and you see much more detailed results showing the most frequently found substances, also with their typical concentrations, and how these substances represent different groups of pesticides, like approved, non-approved, etc. But you can also see the results from the perspective of the countries. So you can go to the results for one of the 11 countries that were involved in SPRINT survey, and you can see the summary of all the matrices, all environmental matrices, human matrices, and animal matrices, and you see the individual substances and the typical concentrations that were detected in this kind of matrix in a given country. And again, after clicking, you will go to the much more detailed results, as I already talked about. The last part of this case study results presentation is the presentation of ecological risks. So that was also shown by Violette. So the values that were measured in the countries in soil and water were then connected to the hazard data, and ecological risk was calculated for air swarms and for fish, for water concentrations. And here we are showing what were the results for the risk quotients in soil for air swarms with the threshold. And you see that in some countries, most of the or many of the measured fields were exceeding the threshold for ecotoxicity. If you click again on the figure, you can see much more detailed results that are also identifying the substances that contributed the most to the overall ecological risk based on the really measured concentrations. The second example I prepared is related to what we call the core of the toolbox, and this is the pathway or cascade connecting using the big data existing across the Europe about the pesticide usage and pesticide application patterns. These data are available from national authorities, and using that we developed the model that is predicting the exposure in the environment, so the concentrations in soil, water, and air. And then again, connecting with the hazard data, we created also the ecological risk maps. So these maps, we believe, can be very useful for the decision makers to identify and design the strategies for pesticide reduction, because we know the overall aims that are 50% reduction of pesticides and 50% reduction of hazardous pesticides. But I think that it's clear to everyone that this is quite ambitious. So I think we need also some kind of prioritization. And our tool is providing the information for such prioritization, because it shows the temporal and spatial hotspots and accumulation of the risk and of the concentrations of individual substances. And based on that, we can then prioritize the substances that at this level show the highest risk or the highest concentrations, and then the measures, like for example changes in crops, can be taken, because also the output maps are connected to the underlying conditions, like for example what crop is growing at which field, etc. So how does that work? We basically mine the information from national registers and national statistics about pesticide use. And with that, we were able to create what we call the GNAP, generalized application patterns. So the typical application patterns, application rates, and conditions of that applications for each crop. Then we used the national statistics that were showing how much is consumed of pesticides in different districts and different geographical units. And using these two information, we created the maps that are showing the load, how much of each active substance is entering each field. So these maps were at the field level, looking like this. In a minute, I will show you also how does that look like in the toolbox. And then by spatial aggregation methods, we created such maps also for the larger regions within 3 model countries, Czech Republic, Denmark, and the Netherlands. And last point was that the concentration values at the field level were accompanied with the hazard data to calculate the ecological risk. I can show you the example how it does look like. So for concentration maps, I can select these field resolution maps, soil, and there is an interactive map where you can select the region. So for example, I will select the district in Czech Republic, soil, and a compound like glyphosate, and I see the map which with individual fields, and I see also the predicted concentrations in this field and what crop was grown there. After the aggregation, we have the country level maps where I can select the country. So for example, Netherlands, soil, some time averaging interval, and one of the 3 model substances. And I see the average concentrations typical for a given region in a country. And with ecological risk maps, it's based on the field level. So similarly, like the concentration maps in individual field, we see the map with individual fields, and I select the cumulative risk, which is summing up the contribution from all the substances that were predicted to occur in a field. And here you see what was the ecological risk in a given field. And you see also which substances contributed the most to the overall ecological risk in a given field. So this is one of the major tools that we really believe can be useful for the decision makers. With another approaches and another models called Pangea and USTOX, also the effort was performed where we tried to do the predicted concentrations in environmental compartments at the pan-European level, and these maps are also available in the toolbox. Another tool example I prepared for today is the tool which is utilizing the already existing data about concentrations of pesticides in food basket within the Europe in different countries, and using also the other big data that are the consumption patterns of the European inhabitants. We were able to produce the maps showing the typical concentrations of substances in food commodities and then also the predicted dietary intakes of pesticides in different countries. Again, I can just quickly show in the map. So it's an interactive map where you can select the different products and substances, and you can see what are the typical concentrations related to different years in European countries. Other tools in the toolbox, which we don't have so much time to show today, is the tool which is calculating the exposure of farm animals, then the interactive presentation of all the ecotoxicity results measured in SPRINT project and all the toxicity results obtained, then the tool which is showing how to assess the health impact on two examples, its effect of glyphosate on metabolic syndrome and glyphosate effect on atrial septal defect. And the last tool is the interactive library of environmental fate models and also fully functional online wind erosion model. So what is the take-home message? We really explored the big data related to pesticides in Europe, and we have to conclude that we have really good and large data around the pesticides that exists, that involves usage statistics or authorization data, crop maps, hazard databases, etc. And we think that these data so far were not fully used, and they really have a very high hidden potential to serve for prioritizations, for decisions, if they are processed the correct way. And we also have demonstrated how to do that in our toolbox. So we think that further improvement can be done if the standardization of this data will be performed and harmonization, because we also found that the data about pesticide usage and data from the national pesticide registers are not very well harmonized, and a lot of data are missing. So we believe that after such harmonization and standardization, the tools like the tools we developed can be really done for the whole Europe, and then the full potential of such tool will be revealed much more than we could show at this moment. So we also created some policy recommendations for the stakeholders. On the first point, that it should be really required that the data related to pesticides usage or registration should be really harmonized. The second, that the tools like we shown, for example, these maps of concentrations or ecological risk, can be very useful to identify and target pesticides and cropping combinations that result in a highest risk for human and environmental health. So that can really help to design the strategies to reach the ambitious goals of farm to fork. And the last thing, that the current hazard risk index 1, which is relatively not well scientifically based, can be perhaps replaced with some better indicators of risk that really consider the spatial temporal changes in exposure, involving also the mixture effects and also involving the novel hazard indicators that were developed within the SPRINT project. With that, I really thank you for your attention.
Thank you so much, Jakub, for your presentation about the toolbox and a little sneak peek of what people can expect. Of course, there will be a second day to really launch the toolbox. I think it's in the chat. It's on Friday, the 13th of February. For now, there are a few questions. For instance, from César. He is asking, can we think of incorporation of IT into the process of the toolbox, for example, AI for creating insights, technologies for monitoring, etc.?
I think yes, why not? It depends. It depends which segment of the model this would be useful. The model itself is not so much complicated that we would need AI for this moment, but it can be used, I can imagine, especially in cases when we have data gaps. So we can use AI to overcome these data gaps to do the simulations of the data like pesticide usage based on the patterns that we know from some data-rich countries. Then we can create, using AI, we can create the realistic patterns for the countries that are not so data rich.
Yeah. Okay, thank you. Yeah. To the audience, officially the time, well, this would be an hour, so we're a little bit over time, but because there are still some questions, and well, it's also nice to have a little bit of Q&A, we decided to let it run a little bit over. So for 5 to 10 minutes, we still have a conversation. If people need to leave, don't feel obliged to stay, just go to your next meeting or whatever. But then we can continue with the people that are interested. There's another question. Could the maps of generalized use of the raw data also be used to derive them? There's a lot of use here. Of the raw data used to derive them to be used by other researchers.
I don't know if I got the question, but what we show is not just the final map of the concentrations, but the code is open code, and we openly also mention the data that were utilized. So especially after the publication of the paper about the model, everyone can repeat. The condition is that you have the right and the right data at the national level with sufficient resolution and the format that is necessary. So that means, for example, for the application patterns, you have to you need the data that are resolute by means of the crop, by means of the active substance, and there should be also the application rates for individual crops, etc. So it's described. The first paper about these generalized application patterns is now in major revision, so I think it will be published very soon.
Good. And do you know if the government pays attention to the insights created by the SPRINT Toolbox?
In our country, not yet. We have some, let's say, colleagues at ministries, and these are, let's say, they were there is kind of evolution. So they were selected because they are interested in what we are doing. So we have these colleagues, and they are usually quite interested to what we are doing, and they are also testing the toolbox, etc. But unfortunately, there are also many others who are not so interested, and even though we are sending them the newsletters or some news about the releases of the data, but I think that's normal situation.
Okay. Yeah, maybe when the launch is there, they start to be more interested than they are now. Then there is also a question to Violette. Could you share your thoughts on developing and setting mixture thresholds for environmental concentrations? You're muted.
Should I share what?
Could
I
you
did not get the...
Could you share your thoughts on developing and setting mixture thresholds for the environmental concentration?
Yeah, sure. We have conducted, especially Jakub and his group, they conducted in the terrestrial environment mixture approaches, and as well we test. What I think the most important thing is that we use mesocosms with multi-species tests to address the real effects, because if you only use single species, we will not find the effects, as I showed, which we can see with mixtures of species. On the other hand, it's important to know that we do not only have additive effects in the ecosystems, but we have synergistic effects. And what we suggest is to take based on monitoring data and recommended applications, these data, to test the effects of these mixtures on the mesocosms. So that means if, for example, for one crop, 10 or 15 different pesticides are recommended per growing season, it would be good to test them on the mesocosms to see what is the changes. And what I was thinking, what could be a good idea to see, okay, we define when no effects, when we have effects of 25% of the population is changing, something like this, 50%, and so on. And what we can as well then define threshold values for pesticide residues in soils before the growing season as a kind of background values. And then saying, okay, if these threshold values are surpassed, then in this season, the farmer should be very careful or have some restrictions to apply other pesticides. But this is something which we will still discuss and publish, I think, in a short term. But these are my thoughts to this. That means take mesocosms with multi-species to test the mixtures based on either recommended application rates or mixtures you will find in field, and test then the effects of these multi-species mesocosms.
Well, thank you for your elaborate thought on this matter. There was also a more technical question. Someone asked, like, SPRINT has addressed 200 pesticides. How much % of the total pesticides used in EU/Argentina represents this? So is it just the tip of the iceberg, or is it already a fair share? I think that's more the intention of the question.
I think that is, look, we have in Europe now about 500 approved. The ratio between approved ones, non-approved ones, banned ones, candidates for substitutions, this is always changing. So we were taking these 200 based on pre-test and as well interviews of the farmers. So we didn't take them all, but I think we took the most important ones. And especially the ones which are not approved, which occur always in all matrices like DDTs, HCHs, and so on. They are persistent of about 50 years. So they are as well taken in there from the non-approved ones. The most important were taken into consideration, and as well from the approved ones and candidates for substitution as well. The ones with the highest frequency were taken into consideration.
Okay, thank you. Then I see one question for Jakub still, and I think this is one of the last questions before we wrap up. To what extent the increase of the bio inputs might create a way out of the agrochemicals? Do you have figures?
That's maybe more general question, not just to me. So this is more related to the transition strategies. Indeed, in Work Package 6, it was explored in the SPRINT project. What are the possible transition strategies? The decisions were based on a large review of the literature available about this problem. And also it was based on the results of the workshops that were run in all the countries involved. It was the set of the workshops, going from the first one, like exploring the possibilities for transition, and then going to more concrete transition steps. And it became quite apparent that change to organic agriculture is only one part of the transition issue. that without the systemic change of the whole agri-food system, the success and substantial reduction of the pesticides and the risk will not be possible. So even though if we will increase, it's definitely helping the situation, but it will not be the final solution to significantly change. I don't know if Violette wants to add something, or Paula, but I think these were the major conclusions from this work package.
Yes, it is. It is. Now it is where the policies in Europe as well have changed. So the farm to fork strategy is not anymore the main strategy or the main goal. So yeah, I think transition is always it's difficult, and we saw as well with farmers that it takes 3 to 5 years. We had these example farmers who showed this as well in Brussels. It takes some time if you want to change the production system, and I think without strong support from governments and financial support in this transition phase, it will be very difficult to come up with a significant transition in the next years.
Oh, well, thank you for your contributions today. I'm wrapping up because we are now 15 minutes over our time. Well, thanks to the audio. Oh, do you want one?
Yes, I just wanted
10
to.
seconds.
Yes, I just wanted to. Ach, jetzt geht das wieder nicht. Warte mal, share. Sorry,
It's
I wanted
shared.
to.
We already see it.
Oh, that's good.
Yeah.
So that's because I wanted, maybe before you wrap up, I would like to say I really would thank all sprinters, as we called ourselves, for the huge contribution you did in all the years. Although we maybe could not contribute so much to the transition of agriculture now in place, I think we strongly could contribute to risk assessments. We could contribute to showing pesticide distribution in the different ecosystems. Jakub with his toolbox, giving a fantastic tool for policymakers, for EFSA, for land use planners to see where are the regions of highest risk related to pesticides, and maybe then as well making some of policymakers thinking in which regions could be other production systems be set into place. So I hope, and I think I'm really grateful for this strong collaboration with this really transdisciplinary team, which was a challenge. It was huge work, and thanks for all of you. And you can see here the colleagues who contributed directly this time to my presentation, but thanks to all other Sprint colleagues a lot. And I was really happy to work with you all these years through, and it was really a pleasure.
Okay, well, thank you for these nice concluding words. If people would like to be updated via Sprint, please have a look at our website. There's so much information out there. There's the publications, there are fact sheets, there are success stories. There will be a link to the toolbox to this webinar. If you search anything related to Sprint, you will find it on the website. So I highly recommend you to have a look. If there are other targeted questions, please send an email to sprint@word.nl, and I wish you all a very nice day ahead, and well, until the next time. Thank you so much.
Bye-bye.
Bye-bye.